Okay, so this segment now is going to focus on fertilization, implantation, and pregnancy. What we're going to cover in this series of notes is looking at the events of fertilization, then we'll talk about early embryonic development and implantation, as well as embryonic and fetal development, and then hormonal changes in pregnancy. So we're going to be covering all four topics. So in fertilization usually, we put usually because that's what the normal is, it's normal for it to take place in the uterine tube, okay, as I've mentioned in previous video lectures. Sperm have to be introduced into the female reproductive tract within 5 days before ovulation. Timing is everything, all right? The sperm are viable for 5 days, and the secondary oocyte is viable for 12 to 24 hours. So the sperm actually, they're initially incapable of fertilization, and they become capable of fertilization through a process called capacitation. So what capacitation is, it's where actually the tail or the flagellum of the sperm moves faster, so that actually increases motility, and the plasma membrane of the sperm is altered, and it's altered in such a manner that it can fuse with the secondary oocyte. Let's just look at sort of a pictorial view of what we were just talking about. So we've already talked about how the secondary oocyte gets produced in the ovary. And we've mentioned that it takes it 4 days to get to the uterus. So while it's traveling through that uterine tube, it may encounter sperm. So the sperm have to enter through the female reproductive tract. So I'm using a blue color for sperm. And the sperm have to encounter the secondary oocyte, and that's where fertilization takes place. Okay, so like I said earlier, timing is everything, right? Okay, so fertilization takes place in the uterine tube. Remember that implantation takes place in the uterus. All right, so here's how this process happens. So we're going to look at sperm penetration of the secondary oocyte. So how does the sperm penetrates? So there's a whole bunch of sperm that actually reach the ovum. Remember that for every ejaculation there's anywhere from 50 to 500 million sperm that get released. So that means a whole bunch of sperm can actually reach the ovum. So once they do reach the ovum, and let's actually write secondary oocyte here because that's actually a more correct term. So when the sperm reach the secondary oocyte, this is when the acrosomal reaction begins. So the acrosomal reaction begins where a whole bunch of enzymes get released, and the enzymes start breaking through the zona pellucida. And so here you can see in step 2, all right, the enzymes that are being released. So you can see how that red area is indicating that. And then we have step 3, breaking through the zona pellucida. Okay, because remember, these have to penetrate into the egg. So now sperm have gained entry into your secondary oocyte. And once they've gained entry into the secondary oocyte, the first sperm to bind to the receptor on the oocyte wins. That's the one that fertilizes the egg. Okay, it's got to bind to the receptor, however. Once that happens, the sperm can get transported to the cytoplasm of the oocyte, and then the zona pellucida will harden, and that prevents what's called polyspermy. So you don't want multiple sperm fertilizing eggs. This picture will look familiar to you once again. So now where we are in this diagram, remember this was the diagram of oogenesis, we are at this level here, after fertilization now. So that is stimulated meiosis 2. Fertilization stimulates meiosis 2. That's where then we go from a secondary oocyte to an ovum. So the sperm plasma membrane then disintegrates. The chromosomes from the sperm and from the ovum migrate to the center, and now we have a zygote. So from that fertilized egg then, the zygote is going to start developing. So remember where we are, we're in the uterine tube. Here's the zygote, which is the fertilized ovum. Now that zygote is going to undergo mitosis. And when it undergoes mitosis, the result is a 4-cell stage. Then it's going to undergo mitosis again, all right, and then it will become a morula. So a morula is literally a solid ball of cells. If you look at this image, it's nothing more than a solid ball of cells. That morula then will undergo more development, and it will develop into a blastocyst. A blastocyst is essentially a hollow ball of cells. Here's a little area that's got some cells, and the rest of this, the blastocele, is actually hollow. The trophoblast will actually, it'll eventually become the placenta, or help form part of the placenta at least. It's at this time that that blastocyst becomes an actual embryo. So with the blastocyst, the reason we're saying that this becomes an embryo is that the cells begin to differentiate. And what I mean by the cells beginning to differentiate is that they're now committed to becoming certain types of tissues. Maybe they're committed to becoming neural tissue, or maybe they're committed to becoming muscle tissue, and so on. Then, finally, here we have implantation at day 6 post-fertilization. And the trophoblast is also very important, or plays a role in implantation. Okay, so a lot of times we get the question, well, how are twins derived? Okay, so if you get identical twins, what's happened with identical twins is that the morula that we talked about on the previous slide, the morula splits. And when the morula splits, you wind up with 2 different embryos, so you have identical twins. Fraternal twins, on the other hand, is where you actually have 2 different eggs that are fertilized by 2 different sperm. So those are fraternal twins. So now we have the blastocyst. So the blastocyst will begin the process of implantation. Okay, so we're in the uterus, and the blastocyst is going to start to implant. So first off, the endometrium, it swells, and that increases, and it increases glycogen stores. Why do you think we're going to need glycogen? Why is that important? Continuing on with this process of implantation, the trophoblast starts secreting enzymes, and that's going to start dissolving away the wall of the endometrium. This is showing how the enzymes are eating away at that part of the wall. We're digesting the cells, and we're providing nourishment for the embryo. At the same time, there's also paracrines that are secreted. And so what that's going to do is it's going to increase the capillaries in the area, so that way you can bring more oxygen and nutrient delivered to the area. You can also then remove more carbon dioxide and remove waste from the area as well. In late embryonic development, so at 3 weeks, you can see what the embryo looks like at 3 weeks. You can see what the embryo looks like at 5 weeks. You can make a comparison there. At 5 weeks, the trophoblast actually thickens. And when the trophoblast thickens, it becomes the chorion, and the chorion encapsulates the embryo. All right, and then by 8 weeks, now we have what we call a little human, right? And now this thing resembles a human, and now it's appropriate to call it a fetus. So at this time, by 8 weeks, the placenta has developed. We're not going to go into details on the exact physiology of the placenta, but the placenta is allowing for exchange of gases and nutrients between the fetus and the mother. Okay, so it's between the circulation of the fetus and the circulation of the mother. Or you could say maternal, all right, maternal and fetal circulation.